very much. I'm Professor Hapuarachchi, is a presenting author of the dual enzyme inhibition and antiglycation activity of the Ayurveda Polyherbal Decoction in type 2 diabetes mellitus. I'm from Faculty of Indigenous Medicine, Department of Ayurveda, Pharmacology and Pharmaceutics, University of Colombo, Rajagiriya, Sri Lanka. So these are my co-authors. Dr. KDC Madhushika, Professor P.K. Pereira, Professor T.S. Suresh from Sri Jayawardhanapura University, Dr. L.A.D.A. Marayawavala from ITI Industrial Technology. So these are the other two co-investigators in Dr. N.D. Kodituaku and Dr. J.M. Dahanayaka. According to the conference regulations, I couldn't add these two co-authors in this abstract. They gave the valuable support to doing this research work. So this is the overview of my presentation: introduction, research gap, objective, methodology, results and observation, conclusion and future work. Introduction. So type 2 diabetes mellitus is a chronic metabolic disorder associated with insulin-resistant and hyperglycemia. Uncontrolled diabetes leads to serious complications such as cardiovascular diseases, neuropathy, and retinopathy. So according to the International Diabetic Federation, there are 589 million adults in between 20 to 79 years living with diabetes worldwide, with 4 in 5 of them, 81%, residing in low and middle income countries. So we are doing some of the work on these through the alpha amylase enzyme. Alpha amylase enzyme is a key digestive enzyme responsible for breaking down complex carbohydrates. Alpha amylase is produced mainly in the pancreas and salivary glands, allowing carbohydrate digestion to begin in the mouth and continue in the small intestine. It acts on dietary starch and glycogen by cleaving alpha-1 4 glycosidic bonds, producing smaller sugars such as maltose, maltose, and other dextrins. As it rapidly converts starch into absorbable sugars, inhibiting alpha amylase is an important therapeutic strategy for managing postprandial blood glucose level in type 2 diabetes. Alpha-glucosidase. Alpha-glucosidase is located in the brush border of the small intestine, especially on the surface of the enterocytes, the intestinal epithelial cells lying in the jejunum and upper ileum. This means the enzyme is positioned right where final carbohydrate digestion occurs. After alpha amylase breaks starch into smaller oligosaccharides, alpha-glucosidase completes the process by converting them into glucose, which is then absorbed into the bloodstream. Antiglycation. Antiglycation activity refers to the ability of a substance to prevent or slow down glycation, a non-enzymatic reaction where sugars bind to proteins, lipid, or nucleic acid. Why is glycation harmful? When glycation occurs, it forms harmful compounds called adverse glycation end products, AGEs. These AGEs damage proteins, trigger oxidative stress, induce inflammation, accelerate aging of tissues, contribute to chronic disease. So we were focused to the Ayurvedic medical formulation. It is described in our Ayurveda authentic text. So in generation to generation, we are practiced in the conventional preparation of decoction. So these are three ingredients: the Sida alanifolia, the whole plant of Pseudalinifolia, fruit bark of Eagle marmalus, and rhizome on Syngivo officinale. What's the research gap? The research gap to identify novel product that could be used for the prevention of treatment of type 2 diabetes. So we were search about effective formulation of Balabilla shunti. This is our decoction name for type 2 diabetes. So whether it is scientific research or validate the our objectives. So for this, we developed the novel pharmaceutical and nutraceuticals from these conventional decoction formula. Our objective of this study was to establish the potential anti-diabetic claim of the freeze-dried and spray-dried form of the hot aqueous extract of Balabilla shunti prepared according to the traditional decoction preparation methods. Methodology. The decoction preparation, the Balabilla shunti decoction was prepared according to the traditional decoction preparation. That means the 60 grams of each raw material mixed with 120 ml of water and boiled to reduce the 240 milliliters. So this decoction we focus to get the dried samples through freeze-dried and spray-dried. This powder was used for investigating the bioactivity studies. Alpha amylase inhibition assay. The alpha amylase solution, 10 microliters, was mixed with the plant extract solution at different concentration. The solution was incubated for 15 minutes. 1% starch solution and pH 6.9 phosphate buffer solution was added and incubated for another 15 minutes. DNSA reagent was added and the reaction mixture was kept in a water bath to terminate the reaction. Absorbance was measured at 540 nanometers using plate reader. Then, alpha-glucosidase inhibition assay. The enzyme solution was mixed with the plant extract solution or acarbose, 300 microliters at different concentration. The solution was incubated for 20 minutes. P-nitrophenyl alpha-D-glucopyronicide 10 micromolecules or 10 microliters and pH 6.8 phosphate buffer solution was added and incubated for another 30 minutes. The reaction was terminated by adding 650 microliters of 1 sodium carbonate solution. Absorbance was measured at 405 nanometers using plate reader. Antiglycation assay. Bovine serum albumin solution was mixed with the fructose solution and the sample was a standard at different concentration. This solution was incubated with phosphate buffer saline solution for 7 days. Protein was used as the standard sample. Blends were prepared only with the plant extract with respective concentration. The fluorescence emission of each mixture was measured with the excitation and emission wavelengths at 355 nanometers and 440 nanometers respectively using fluorescence spectrometer. Calculation. All the tests were carried out in triplicates for alpha amylase and alpha glucosidase assays. Clinically used drug acarbose was used as standard. For antiglycation assay, routine was used as the standard. Percentage inhibition was calculated using the following equation. Percentage inhibition is similar to control minus plan versus control into 100%. Graph of percentage inhibition versus concentration was plotted to calculate the half maximum inhibitory concentration. Statistical analysis used for these studies, results were expressed as seen plus or minus standard deviation. 1 way ANOVA was performed and the p-value was less than 0.05. Minitab software was used for data analysis. Results and observation as like this with the ACABOS standards. This is freeze-dried Balabilla shunti, and this is the spray-dried Balabilla shunti. These are IC50 values is 4.5 plus or minus 0.07 milligram per milliliters. Freeze-dried Balabilla shunti is IC50 values 5.01 plus or minus 0.17 milligram per milliliters. Alpha-glucosidase studies percentage inhibition and IC50 values. With the ACABO standards, their IC50 value is 454.23 plus or minus 1.89 milligram per milliliters. These are the freeze-dried Balabilla shunti one. And it is IC50 value is 2.82 plus or minus 0.09 milligram per ml. Here we are showing this spray-dried Balabilla shunti. IC50 value is 2.38 plus or minus 0.09 milligram per milliliter. Antiglycation study, percentage inhibition and IC50 value. The standard is routine. IC50 value is similar to 316.78 plus or minus 12.143 milligram per ml. Their freeze-dried Balabilla shunti shows IC50 values 445.86 plus or minus 8.66 microgram per milliliters. Spray-dried Balabilla shunti IC50 value is 528.36 plus or minus. So, conclusion of this one. Actually, before that, I want to mention that Ayurveda medicine presents a diverse array of formulation for the geriatric conditions, such as the Balabilla shunti decoctions. This decoction comprises of root bark of eagle marmelas, rhizome of rhizome of Zingiber officinale and whole plant of Sida codifolia. Despite the utilization of Balabilla shunti within clinical settings, it has not been comprehensively explored from a scientific perspective regarding its nutraceutical potential. Use of plant-based nutraceutical with polyherbal formulation is in high demand in developing and developed countries, and they are more effective than purified compounds due to beneficial synergistic interaction of the components in polyherbal formulation. We observed these scenarios and aim to develop Ayurveda polyherbal nutraceutical and to evaluate the claimed bioactivities of different dosage forms. Furthermore, we plan to accomplish our objectives through developing a quality standard Ayurveda polyherbal nutraceutical with the scientific advancement of the existing formula as a ready-to-use commercial value-added product. It would be lead to initiate future collaboration with local and international nutraceutical industries, as well as create the attraction of foreign investors. Ayurveda and traditional system of medicine in Sri Lanka generally practice the use of herbal or herbal mineral nutraceuticals for the management of chronic diseases condition as well as for the general well-being of people since infant period. So with the conclusion of this study, we can show this Balabila Shunti deposited dual target inhibitory activity against uh, alpha amylase and alpha glucosidase enzymes, along with anti-glycation activity supporting its traditional antidiabetic use. Due to the potential of Balabila Shunti decoction to act as an alpha amylase inhibitor, it may serve as an effective herbal therapy in management of diabetes. Future work, because of due to limited time duration and the unavailability of the further funding sources due to the financial crisis in Sri Lanka, we couldn't identify the isolation and identification of biomolecules with antidiabetic activity from the traditional Balabilla-Shunti decoctions. So these things and other also investigation of methods to incorporate isolate biomolecules to nutraceutical and pharmaceutical products to develop effective products. These works we are supposed to do in our future work because these works are essential to standardize the value added product. In future, we hope to commercialize this value added product for the type 2 diabetes mellitus patient as an elderly nutraceutical product. These are the references here. Thank you very much for my presentation.